Hello everyone and welcome to my YouTube channel Perfect Lab. Have you ever wondered why microbiologists incubate bacterial culture plates upside down instead of upright? It may seem like a simple step, but it plays a crucial role in obtaining accurate and reliable culture results. In this video, we will explore why bacterial plates are inverted during incubation, how condensation forms inside a Petri dish, and the problems that may occur when plates are incubated the wrong way. So let's get started. The main reason why bacterial culture plates are inverted during incubation is to prevent water droplets from falling back onto the agar surface as shown over here. This is because during incubation, that is around a temperature of 35 to 37 degrees Celsius, moisture condenses on the lid of the Petri dish as shown over here because of the warm temperature of the incubator. So if the plate is kept upright, the water droplets can fall back onto the agar and cause several problems. And this way of incubation leads to higher risk of contamination in your microbial work. So the best and the recommended way to prevent water droplets from falling back onto your inoculum on the culture plate during your incubation is to invert or turn the bacterial culture plate upside down during the incubation process as shown over here. And this way of incubation will prevent your microbial work from contamination and ensure a good, reliable lab result. So the key point here is that inverted incubation prevents water droplets from falling back onto the agar surface. And this, as a result, keeps the bacterial colonies purely isolated and uncontaminated, leading to a reliable and a more accurate result. Let's look at the problems caused by upright incubation of bacterial plates. The first is spread of bacterial colonies. The first picture here shows an inverted incubation of a bacterial plate, and the second image or the picture here shows an upright incubation of bacterial plate. So our focus is going to be on the upright incubation of bacterial plates. So during incubation, water droplets formed by condensation on the lid of the Petri dish as shown here can fall back onto the agar and carry bacteria across the plate. And this causes isolated colonies to spread and merge, making it difficult to differentiate, count, or isolate individual bacterial colonies as shown here. So the second problem is contamination. And again, the picture over here shows an inverted incubation of a bacterial plate, and the second image shows an upright incubation of a bacterial plate. So condensation can also increase the risk of contamination during upright incubation as shown over here. This is because during incubation, water droplets may form on the lid of the Petri dish. And if these droplets fall onto the agar surface, they can pick up microorganisms from one part of the plate and carry them to another part. And as a result, contaminants or unwanted bacteria can spread across the plates. And this makes it difficult to determine whether the observed colonies, as shown over here, originated from the original sample or from contamination. The third problem is distortion of results. And over here, what we have to note is that excess moisture formed on the agar surface as a result of upright incubation, as shown over here, can also distort the normal appearance of bacterial colonies. Example, instead of bacterial colonies growing as well-defined round colonies, bacteria may spread over the wet surface, causing colonies to become irregular in shape, larger than expected or even merge with neighboring colonies. And this abnormal growth makes it difficult to accurately observe colony characteristics such as bacterial size, shape, and elevation, which are important factors for bacterial identification. The fourth problem is agar surface damage, as shown here. And again, too much water can make the agar surface wet, soft, or uneven. And this also interferes with the normal bacterial growth and the even diffusion of antibiotics, which can affect the accuracy of the test result. So thanks so much for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.